My name is Daniil Gataulin. I'm one of senior research analysts covering biotech here at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. It is my pleasure to introduce Laurent Fischer and Rick Beckman from Adverum to our virtual ophthalmology summit, where the initial focus is on wet AMD and diabetes-related ocular diseases. Laurent, Rick, welcome.
Thank you, Daniil. Pleasure to be here.
Great. To start off, can you give a brief overview of Adverum and its program?
Of course. So Adverum is focusing on highly prevalent ocular disease, and our most advanced program is a gene therapy that is an aflibercept encoding AAV27M8 vector that is the benefit of being administered intravitreally as an outpatient basis. We are in the clinic with a phase 2 study called the LUNA trial, and we've already demonstrated with our Ixo-vec vector that past 2 years we could actually stabilize the fluid in the patients with wet AMD and maintain vision in patients who require frequent injections. We believe that is a significant medical need. Our 7M8 platform is also being used in other programs by GenSight, was recently licensed by Ray Therapeutics as well. And again, one of the benefits is does not require a surgical procedure to administer this gene therapy product.
Excellent. Thank you for that. And I guess we can jump right into it. And you mentioned the ongoing phase 2 LUNA trial. Can you talk about that trial? Maybe dive a little bit deeper into the trial design.
Rick, would you like to talk about the LUNA phase 2 trial?
Sure. The goal of the LUNA trial is twofold. One is to determine the best dose, and the other is to also determine the best prophylactic corticosteroid regimen. It's become very well known in the field that when you put trillions of viral vectors into a patient's eye, that you are stimulating the immune system. And what we've learned is that's very much dose related, and we believe that the best method of preventing any inflammation in the eye is an adequate prophylactic corticosteroid regimen. People who were not using it in the past are now using it. A lot of people who've been using it all along are now admitting that they're using it. We feel that we have a tremendous amount of information about that. But what we want to determine is the best regimen that we can do. And so we're looking at 2 different local regimens. One is drops and the other one is injectable polymer. And then we're also looking to see whether we also need a systemic coverage, because people believe that there might be a systemic component to the inflammation seen in the eye. So we do 2 different locals, both with and without a systemic corticosteroid regimen. And then we're looking at 2 different doses. We're looking at the 2E11 dose, which we showed in the OPTIC clinical trial had tremendous efficacy and a very good safety profile. But our preclinical studies have allowed us to determine that we have a very flat dose response curve. And indeed, looking at the preclinical aflibercept levels of non-human primates, we see that we can get essentially the same levels we believe with the 6E10 dose. And because we have this flat dose response curve that you can see in the left in terms of 3 different log units of aflibercept levels are essentially equal, going from the 3E10, which in the non-human primate is equivalent in the human to 6E10, all the way up to 2E13, we believe that based on the fact that inflammation, as you see on the right, has a rather steep dose response, that by dosing down, we can maintain the same aflibercept levels, but have less of a stimulus for inflammation. And thus, as you see, going from the light yellow box to the darker yellow box, we're expanding our therapeutic window by dosing down. So the design of the LUNA study is to look at 4 different prophylactic regimens and to look at 2 different doses, one being the 2E11, which was the low effective dose in the OPTIC study, and then the potential for an even lower 6E10 dose.
Got it. Thank you. And in light of this design, and I guess together with the phase 1 data, how should we be thinking about the success of the phase 2 trial?
So I think, if you look at the newer treatment options that are available, about half of the patients get to 16 weeks treatment with Vabysmo and with Eylea 8 milligram. And that's clearly an improvement upon the existing treatments. But what we've seen in OPTIC, as Rick was referring to, is that half of the patients at 2 years had 0 injections. So a single injection was able to maintain the fluid out to 2 years. And we saw more than 80% reduction in annualized anti-VEGF. So that's clearly superior. I think the other thing that's really more important is that when you have, the current treatment regimen, you see a lot of fluctuation in the central subfield thickness. We now know that that fluctuation leads to poor long-term vision outcomes. And what we see actually in an OPTIC study, and there are a couple of cases that maybe Rick can walk us through, is that we have really a great way to actually stabilize the retina and actually keep patients without fluid. And if I may share just a couple of slides, I think it's very clear in some of these cases studies that we can really improve the current standard of care by really offering something that's different. And maybe Rick can go through these kind of 2 cases, because I think they make the point very, very clearly.
Yeah, thank you, Laurent. So, the point is that it's not just the presence of fluid or the absence of fluid, but the most recent data that's coming out of Europe and other places in the United States is that it's actually the fluctuations in fluid, and that's caused by bolus injections. They have the half-life of Eylea is about 7 to 10 days. And so what happens is when you give the injection, the fluid initially goes away, but over a period of time, as the Eylea starts to wear off, you start to see a reaccumulation of fluid. The other thing that happens is a patient misses a visit and you get accumulation of fluid. And they've actually shown it's the fluctuations in fluid that leads to long-term vision loss. So what you see here is a patient that's treated at one of the best retina clinics in the United States. And indeed, they got more than the average number of injections in the past year. They got 9 injections. Each of those light green blue dots represents an OCT reading and an aflibercept injection. And so what you're seeing is that there's a fluctuation in the amount of fluid in the retina. The CST is the thickness of the retina measured by OCT. You see it going from 400 to 300 back up to 400. And then at negative 30 weeks, what you're seeing is a missed injection between negative 30 and negative 21. And you can see there's a tremendous accumulation of fluid. And normally patients, they go away on a vacation, they have to miss an appointment. And when you miss an appointment, even by a couple of weeks, you see an accumulation of fluid that can occur. Now, this particular patient received their Ixo-vec injection with the dotted line, and each of the open circles represents an OCT measurement of thickness. But what you're seeing is it's very, very stable down around 200 microns, and that there was no need for any additional injections. And so what you see is this sawtooth pattern of fluctuating vision or fluctuating fluid in the retina. We call that fluid volatility, has now gone to a very stable, non-fluctuating, which we believe is going to represent preservation of vision over the long run. And so what you can see is this particular patient actually gained some vision at the end. They gained 13 letters in visual acuity, despite the fact that they were already being treated rather frequently with intravitreal injections. If we want to show the next slide, and I'm going to point to the OCTs on this one because they're easy to see. If you look at the top left, you see that there's areas of black within sandwiched areas of white. The white is normal retina. Those areas of black encapsulated in there is intraretinal and subretinal fluid. Now, looking down at the OCT, you can see that this particular patient, despite 9 injections of aflibercept, they maintained almost 650 microns of intraretinal fluid and subretinal fluid. This is something that is not being very well treated, despite the fact that they're getting multiple injections. Now, if we show the next slide, you can see that they received their Ixo-vec administration, and afterward, you can look at the OCT. Those vacuous spaces there are now gone. You can see that there's no longer any fluid on OCT, and the patient has not required any injections. So what we're seeing in this particular patient is that perhaps the benefit of getting steady state production of aflibercept and neutralizing any VEGF that occurs over the long run is actually not only leading to lower volatility in fluid, but actually a significant improvement in the amount of fluid. This patient was a tremendous responder.
Got it. Understand. And maybe along the same lines, we do see those measurements from other companies, other trials as well, but not a lot of commentary around the clinical significance of that. Can you provide some commentary around how clinically significant those results are?
Yeah. So, one of the paper authors was Usha Chakravarthi from the UK, and she told us that she believes actually that it's the actual mechanical deformation of some of the cells that bridge the layers of the retina that's causing the ingrowth of fibrous tissue, scar tissue, and leads to atrophy or lack of tissue in the retina. And so we really believe that this decrease in the volatility of the fluid, which can only be done by something that provides for the continuous production of aflibercept. So even if you go from Eylea every 8 weeks, out to Vabysmo at every 12 weeks, or maybe even out to every 16 weeks, there's still a waxing and waning because of the bolus method of injection. And as people try to expand that treatment interval, you're going to see even more of that, because expanding the treatment interval is associated with the loss of efficacy of the drug. At some point, you're really treating until they fail, and then you're backing off a little bit. The difference between going out to every 8 weeks or every 16 weeks and maybe once every year, or maybe never another injection for the next 10 years of your life, it's an order of magnitude difference in terms of what the clinical response is, not only in terms of the burden, the treatment burden, which is really, really a very significant component to this, but we really believe in the long run, the maintenance of visual acuity is going to be the key to this. And we believe that that's why the Europeans saw the value and gave us prime designation, not just based on the treatment burden, but they're losing vision over there despite having to pay for multiple injections. We think that this way we can preserve vision.
Got it. Understand. Yes, go ahead.
So this is, the label of Vabysmo, and I think it's very clear that it works as well as Eylea every 8 weeks. It's still a number of injections, 10 injections, 15 for Eylea every 15 weeks, but and a reduction certainly the second year. But you can still see these fluctuations in CST are pretty significant. And I think that's, again, in the best case scenario in a clinical trial leading to the approval of a drug. When you look at the real world, a lot of patients cannot actually get their eyes injected as frequently, so they do lose vision. Some of them stop growing altogether. So I think the potential for these two patients, essentially, it's a functional cure. And what we've shown in our trial is that we can measure levels of aflibercept at 10 to 14 weeks, and they predict essentially as far as we measured it, which is 3 years. And so we'll present later this year 4-year expression. We expect that for these patients, this would be a lifelong benefit. So this is, again, the longer we follow these patients, the better the benefit risk, the greater the benefit. And for some of these patients, in fact, half of them that receive that low 2E11 dose that we're testing in the LUNA trial, it's a functional cure. They no longer have active disease, they no longer have fluid in their macular retina.
Got it. And to put all this into, into practical context, what have you been hearing from both the physicians and the patients?
Well, one of the things we hear from some of the physicians and their patients is they want to know if they can have it done in their other eye, because now some of these people, a lot of them are bilateral disease. There's about a 10% conversion to bilaterality per year. And so what the patient realizes now is, hey, I'm not getting injections in my one eye. This is life-changing. It would sure be nice to be able to do it in the other eye as well.
I think for some of these patients were able to go back to work because they actually were able to see that their vision was really stabilized and not fluctuating anymore. In some cases, these patients could go back to work. Most of them are retired, but they all have daily activities, and this is really life-changing for many of these patients.
Got it. Understand. And Rick, and to your point, I guess to take it one step further, where are you at in terms of going into the second eye?
So we haven't talked about that publicly, and I won't make any comments. But what we have seen in our preclinical studies is the proof of concept that even though we generate some neutralizing antibodies systemically after a first injection, we do not see any neutralizing antibodies in the contralateral eye. And that gives us good thoughts that allows an open door for being able to do that. But we'll be looking into that in the future, and we'll be talking about it in the future.
Got it. That's fair. So is there, do you think, a level of anti-VEGF injection reduction annually that you believe physicians and patients are looking for when deciding whether to take this gene therapy?
So I think, what we hear consistently is that a 50% reduction in annualized anti-VEGF would be very meaningful. We've seen, 83% in optic at 2 years. So that's quite better than what we think is the threshold, and 50% entirely free of injection. And I think that's kind of a best-in-class effect for anti-VEGF and wet AMD treatments. So anything in that zip code, I think, obviously would be great. Anything above 50%, I think, would be readily accepted, as long as the safety profile looks good. And that's why we've been focusing on optimizing the prophylaxis to have a reliable, predictable prophylactic regimen with only a very small fraction of patients requiring post-prophylaxis management.
Okay, got it. And regardless of how efficacious gene therapy approach is, we can all agree that it's not for everyone. So I guess a couple of questions here. Who do you see as ideal patients for your approach? And in a scenario where the therapy is approved, what would you envision the potential launch here to look like?
So a couple of thoughts. I'll let Rick answer, but just a couple of thoughts. If you think about, the state of treatment of wet AMD in the US, the IRIS registry that follows 2 million people, the data they presented that's a little bit over a year ago showed that only 60% of the patients received enough injection. That was 8.8 per year, which is almost 1 every 5 weeks, which is what we've seen as the population that was enrolled in both optic and now in the LUNA study. So essentially, a majority of the patients would benefit greatly from being able to maintain their vision and reduce dramatically the number of injections. And that's why we received PRIME designation in Europe last summer, and more recently this year, we received ILAP designation in MHRA. It's the only program that received Fast Track, ILAP, and PRIME. And the reason for that is we can really dramatically improve the standard of care, in this disease that is well treated with a very high treatment burden that's not really attainable by many patients. In fact, less than 60%. So there's a lot of room for improvement. Okay. Sorry, Rick.
The way I like to look at it is if you were to ask 10 patients if they'd like to get an injection in their eye with a needle, what percentage of them would say that they prefer to do that? I don't think anybody likes getting injections in their eye. My mother gets injections in her eye every 8 or 9 times a year. She'd like to be able to move away so she's not so close to a retinal specialist. I think initially patients who get frequent injections, but I think ultimately it's for anybody that gets injections in their eye, because most people need to do it on the order of 8 to 10 times per year. And I don't think anybody likes having to do that. The other thing I call your attention to is that even in that, we saw that more than 50% of the patients, it was one and done at 2 years, meaning that they basically had functional cures. But even for that group of patients that does require some injections in the future, remember, they still have a baseline level of aflibercept being produced by their ixavet. And even if it's not quite enough in that subset of patients that they no longer need any additional injections, it still gives you a baseline protective level for that time when maybe they missed their visit, keeps them from reaccumulating fluid. And remember, people who go from Eylea to Vabysmo, it's with the idea that maybe they can go from every 8 weeks to every 16 weeks. That's having the number of injections or a 50% reduction. I think that most people wouldn't mind taking eye drops for a few months if it gave them the ability to never need to have a needle stuck in their eye again. I think it's people do it so frequently because it saves their vision. But back in the days before anti-VEGF injections, people used to say, who's going to let you stick a needle in the eye? And the answer is they'd rather do that than go blind. But I think that they'd much rather have one and done than have to get injections done every month for the rest of their lives. So I think it's for anybody who requires anti-VEGF ultimately.
But to add a little bit to your question about adoption, I think what's important to have a readily adopted gene therapy product is to have that administered on an outpatient basis, because that fits into the current practice models. We know that these practices are really busy. They see 70 to 90 patients. If they need to do a subretinal surgery that takes an hour, that's really much more challenging. I think suprachoroidal approach still has some technical challenges or training that's associated with it, probably better, but intravitreal injection is really the gold standard for the treatment of wet AMD. And I think that's what we're pursuing with Ixo-vec. It allows really to fit in the current model of the practice model, including their buy and bill model. And I think those are all factors that we're trying to better understand to make sure that we really can support the adoption of a product that could have a huge impact both on the physician practices. It's also important to know that we hear from the adoption and approval of dry AMD treatments, as well as the aging of the population. There are not enough retina specialists, so there's a huge amount of constraint on even just delivering those injections. So we think that we are offering a solution that could be not only cost effective, but maybe cost saving as well, because as you probably know, 12% of Medicare Part B is actually spent on the treatment of wet AMD patients.
Got it. Thank you. And as you elaborated earlier on efficacy, 80% reduction in supplemental injections, and with over half of those remaining injection-free with the 2E11 dose. A couple of questions to follow up on here as well. First, can you elaborate a little bit on the exclusion-inclusion criteria, and how do they compare with the other gene therapy trials for wet AMD?
So we don't go into great detail about our inclusion-exclusion criteria. What we do say is that they need to be 50 years or older. They need to be people who have responded in the past to an anti-VEGF agent, because you're putting gene therapy in patients. And those are the major criteria that we have. Looking at all the other ongoing clinical trials, they're ongoing in a lot of the same sites. And I would say that the inclusion criteria between most clinical trials are rather similar.
Okay. And with respect to the two doses that you're looking at, 6E10 and 2E11, do you expect to see a dose response between those?
Based on the non-human primates, not really. Maybe a little bit. It's really hard to say for sure. We think that with the 2E11 we had excellent efficacy, and if we stay within that similar range, we think that it'll be very helpful. The whole idea with the 6E10 is to not have a dose response in terms of efficacy, but maybe have a little less of an immune response, which we think would be helpful. But we really believe that the efficacy and safety that we had with the 2E11 is very compelling for going for an approval. We think we can do better because, as we've talked about before, the OPTIC study did not have an adequate prophylactic corticosteroid regimen. We know much more about it now, and we believe with an adequate prophylactic corticosteroid regimen. This is just showing some data on non-human primates between the 6E11 and the 2E11 dose. You see that they're half, because the doses in non-human primates will be the 3E10 and 1E11. We don't give any prophylactic corticosteroids to the non-human primates, because we want to see the full range of an inflammatory response if one is present. You can see with the 1E11, which is equivalent to the 2E11, that only half of the animals had any inflammation, and it was relatively short-lived. But when you step down into the 3E10, you can see that it's very significantly less. Only one animal had very mild inflammation. And so learning from this, and based on what we believe will be our therapeutic window, we believe that stepping down to 6E10 is likely to give us essentially equivalent aflibercept levels, but with less stimulus for inflammation.
Okay.
And
yes,
that's fairly unique to our program. We've seen in other programs that as they dose down, they've seen lower levels of aflibercept and less efficacy. And so there's a point where it drops down. It's unique to each capsid and each vector. And what we will be showing in the third quarter is levels of aflibercept at 14 weeks to confirm that what we've seen on human primates, which so far has been confirmed in humans across the entire program and every dose we've tested. But we want to confirm that. That would give us great confidence that 6E10 dose is likely to be as effective with less immune response or immunogenicity than the 2E11.
Got it, understand. And I wanted to talk a little bit more about the supplemental injection criteria. I understand that especially during the earlier trials, this could be, the reason for some variability there. Can you talk a little bit about your supplemental injection criteria in the phase 2 trial, and if you can comment on how that compares with some other ongoing trials?
Yeah, in the OPTIC trial, the first injection had strict criteria and had to be met by a reading center, and then afterwards, any other supplemental aflibercept injection was at the discretion of the investigator, and we determined that that's probably not the best way to go. So what we decided to do in this study is to make all measurements that determine whether or not they're going to get supplemental aflibercept at the discretion of the reading center, following very strict criteria. The criteria are pretty standard across the industry. They've been discussed with our scientific and clinical advisory boards, and everybody feels pretty confident that they're designed to look at the efficacy and safety of the clinical trial while keeping patient safety at the foremost. And so it's pretty standard across the industry with what people use as the actual criteria.
Got it, understand. Okay, wanted to jump and talk a little bit about safety. And first of all, I think probably the biggest overhang that I keep hearing is how the data from your DME trial, the safety signal from your DME trial, translates into wet AMD. Any learnings, things like that you took into consideration.
Maybe before Rick gets into more of the details, I would say that there are a lot of learnings from this program, including the fact that, as I mentioned, there was no drop in efficacy from non-human primates to human, which ended up being the first dose we tested. Normally, you dose escalate. We actually been dose de-escalating since that first dose. So that dose was too high and gave us 100% efficacy, but with some inflammation and some safety signals. We've learned a lot and we've done more work. You've seen actually the reduction in doses leading to still the same levels of aflibercept with much less immune response. And I think that the safety in that very classic dose-limiting toxicity is in the rearview mirror, and we fully understand what happened in this particular study with the 6E11 dose in patients that were not adequately prophylaxed and had, microvascular disease. But, we can talk about it. I think, for me, this is really the importance of having these programs being followed, and we follow patients up to 5 years, and we have now, long-term data. We present 3 years data on OPTIC at 2E11, 6E11, where we haven't seen any such case where, once we understood how to identify them, and we were able to really prevent those patients from having these complications. But, Rick is the expert. He was actually a glaucoma specialist, so he can talk a little bit about what we've seen and why we believe that we can treat it, prevent it, and won't see it in wet AMD.
I think the most important thing I'd like to say is we believe in transparency. And as a result of this, the FDA knows all of the details regarding the Infinity study and the dose-limiting toxicity, and they gave us permission to move forward with our LUNA study based on all of that information. And the European regulators have all of that information as well. I presented all that information to them, and their basic comment is, hey, you got great efficacy, you got to figure out your dose and your adequate prophylactic corticosteroid regimen, because I think the regulatory agencies and industry in general are now coming around to realize that when you put viral vector in an eye, that you stimulate an immune response, and it's dose related. And so what we did is we did a tremendous investigation of what happened in the Infinity study, and we continue to follow those patients. And I'll also add that the doctors that treated those patients in the Infinity study are currently our investigators in the OPTIC study, in the LUNA study, and they are very vigorous in enrolling patients in this study. So the regulatory agencies and the investigator community realize that we have a handle on what was going on. And I'm just going to briefly explain to you what went on. And it was a patient in the 6E11 dose that did not have an adequate prophylactic regimen. In defense of the people that were running the study then, they did not know what the adequate prophylactic corticosteroid regimen was, and they went a little bit lower than I would have. However, the retrospectoscope is 2020. The other thing is that a patient developed early signs of inflammation that were very mild and subtle, that the community didn't really know was a harbinger that something potentially was going to happen. And by the time people realized that these very mild signs of inflammation were ongoing in these diabetic patients, the patient developed one of the sequelae of inflammation, which is hypotony. And the reason that the patient developed this low pressure in the eye is because they were a very brittle diabetic patient. They had tremendous microvascular disease, is what you see with diabetes. And people with diabetic microvascular disease have microvascular problems associated with their kidneys. They have it with their joints, with their extremities. They get amputations. This patient had heart issues as well. And what they were doing is they had microvascular involvement of their ciliary body, which is what makes the fluid in the eye. You're constantly secreting fluid and constantly draining it, and that keeps the pressure normal in the eye. Diabetics like that don't make much fluid. And so when you combine the inflammation of the ciliary body, which occurred in that patient, the fact that it wasn't picked up early enough and treated aggressively enough, that patient developed a decrease in the secretion of aqueous and an increase in the outflow from the inflammation. And what happened is they developed a very low intraocular pressure. Now, we had two patients afterwards which started to develop this, but because people picked up on the early signs of inflammation and treated them very aggressively for inflammation, they never developed that profound hypotony that resulted in vision loss. And so in this particular very at-risk population, a patient that was not diagnosed early enough and treated aggressively enough and wasn't prophylaxed appropriately, that received a very high dose, had a very significant dose-limiting toxicity. The way that you deal with that is, one, is you don't need to use that 6E11 dose. Is two, we're not going into this very at-risk diabetic population with all of the vascular changes that put them at risk for developing low pressure in the eye. And finally, and most importantly, we've learned that you do need to use prophylaxis. The community accepts that you need to use prophylaxis. And an ounce of prevention is worth a pound of cure. So in wet AMD, we're in a different patient population. We're no longer pursuing the 6E11 dose. We're using the 2E11 and even lower 6E10. But we've developed very well-tuned prophylactic corticosteroid regimens that take into account maximum immune suppression during the period when there's peak exposure to the immune system of the viral capsid. And then we wean them very slowly over a period of time, as recommended by our experts, to prevent the occurrence of any inflammation. And we feel confident, the regulatory agencies feel confident, and the investigator community feels confident that with these things in place, that we can safely treat patients in wet AMD. And that's why Luna is enrolling very well.
And to add to what Rick said, just, we did receive PRIME designation and ILAP well after we had our dose-limiting toxicity. So these regulatory bodies have seen all of the information as it relates to safety.
Got it, understand. And speaking of those prophylactic regimens, I believe you're looking at 4 different ones. Can you tell us what they are and how did you design them?
So the way they were designed is, we saw from our preclinical studies in non-human primates that the period of maximum inflammation was about 8 to 12 weeks. And we believe that the period of maximum exposure to the immune system is earlier on, because your exposure leads to inflammation later on. And so what we designed was regimens where during the first 5 or 6 weeks after exposure to Ixo-vec, the administration of Ixo-vec, that we want to treat them with maximum immune suppression. And then what we do is we wean slowly over a period of time until we get the patient off of any anti-inflammatory drugs. In some cases, out as far as 20 weeks. In some cases, it's a little bit different regimen.
Got it. Understand. And I guess speaking of inflammation, what do you view as an acceptable level, if any?
I think it has to do with a couple things. One is it has to be predictable. So what we want to do is we want to predict that the majority of patients after their prophylactic corticosteroid regimen require no more anti-inflammatory agents to treat inflammation. Remember from the OPTIC study, even with inadequate prophylaxis, and I would say very much inadequate prophylaxis, fully 40% of those patients after their mild prophylactic regimen never required any treatment with corticosteroids afterwards. So the idea in mind would be a predictable number, small number of patients that require some additional topical corticosteroid therapy after their prophylactic regimen, where we can where they can be treated predictably, that it goes away, and also that it's easily detectable, and we know that we can detect it now. We know exactly what to look for. So I would say a small percentage of patients that require post-prophylactic treatment of inflammation that's mild and amenable to something such as topical corticosteroid therapy would be a very relevant safety profile.
Got it. Understand. I see we're getting a little bit short on time, but I had a couple of other quick questions. First, you mentioned a couple of licensing deals that highlight your capsid validation. Can you talk a little bit about those?
Sure. We have 7M8 in two actually clinical programs. One is Genzyme GS030 program that's in the clinic, which has demonstrated both efficacy and safety, in addition to Ixo-vec, our own program with AMD. We also have recently licensed 7M8 to Ray Therapeutics, and it's a company that's developing gene therapy products for RPE, so for rare diseases. The CSO was a former co-founder for DMT, and we're very excited to actually, partner with other companies to actually make our technology available. It is the best intravitreal capsid. It's been involved in multiple species. It always shows up as a top screen in the retina, and it really has the most data. And so far, there hasn't been any capsid that shows better, basically transduction of the back of the eye. And what's unique about 7M8 is that loop actually speaks to a loop that actually allows the vector to cross the ILM, the inner limiting membrane. That has actually been, one of the challenges in gene therapy is the, fact that most of them has to be delivered subretinally or now suprachoroidally, which is also a challenge.
Got it. Understand. And I guess to live on the last question here, what do you think investors are missing or not fully appreciating about Adverum's story?
Well, I think, investors are getting more, knowledgeable about gene therapy, the various programs. Obviously, some are more advanced, but have some challenges. We talked about the delivery routes, and then are looking at, alternative routes, which seem to require more vector and have, not as significant inflammation as well, require prophylactic therapy. Others are very early, and I think they want to see that the data we will be sharing confirms what we believe, which is we can actually move forward with a dose of 2E11 or 6E10. If we can move to a dose of 6E10 with the right prophylactic regimen and have manageable safety, then it's clearly a huge opportunity. I think there's been challenges in both ophthalmology with some programs that have failed, as well as in gene therapy. And I think we've been involved in that number of companies that had some challenges. I think we've learned and had more experience than any other company in the space, and have the most attractive efficacy and safety profile with the 2E11 dose. We're improving upon that. And hopefully we are educating investors about this opportunity. And the fact that we have essentially an entirely new team that is very experienced in gene therapy, with multiple involved in actually getting gene therapy program and ophthalmology approved around the table, a lot of experience, and that we have the funds to see through the data from our phase 2 study to help identify a partner for ex-US, co-development for this multi-billion-dollar opportunity. And I think the fact that people are starting to realize that, Eylea, aflibercept still, is the best anti-VEGF out there. Sure, Vabysmo is as good, but not better. And Eylea 8 milligrams seems to be very competitive. We think that delivering aflibercept at low levels permanently is the best way to actually treat patients. And that is a significant benefit for all stakeholders, patients, as well as investigators and the healthcare system. So this is a really unique win-win-win situation, and we're excited to share the data in the third and fourth quarter this year, talking about pivotal trials, in the upcoming months, and giving more guidance to when we believe we can start those trials, and with vision to BLA filing, and potentially in Europe with a prime designation. There's a path to more accelerated approvals, and those are things we'll be discussing with EMA, which has been a bit more receptive to gene therapy than their US counterparts in some cases. So we're very excited about the program and look forward to continue to give updates to investors and make them aware of this opportunity.
Excellent. Thank you very much for that, Laurent. And again, thank you very much for joining us. We are out of time now, but always great speaking with you, and look forward to our next chat. Thanks, Laurent and Rick.
Thanks. Great speaking with you.
Great speaking with you. Take care. Bye-bye.
Bye-bye.